A healthy immune response acts like a finely tuned electrical impulse. It spikes to neutralize a threat, then smoothly decays back to baseline. But in certain systemic conditions, that signal fails to decay. It morphs into a self-reinforcing, chaotic runaway wave. We recognize this clinically as a cytokine release syndrome or a cytokine storm. It is a catastrophic loss of negative feedback control. In severe conditions like viral pneumonia or acute respiratory distress syndrome, the physical ground zero for this system's failure is the alveolar capillary barrier of the lungs. To understand why this barrier collapses, we have to look at the primary biological engine driving the loop, the receptor for Advanced Glycation End Products, or RAGE. RAGE functions as the innate immune system's central amplification node, acting as a physiological hub where diverse tissue stress signals converge into a singular, runaway inflammatory response. To map exactly how this happens, we need to deconstruct RAGE from the angstrom level up. We will analyze its structural permissiveness, its pathological genetic loop, and its receptor crosstalk. Examining the molecular architecture of the RAGE-ligand axis provides the necessary framework to understand how a localized infection scales into systemic organ failure. Zooming in from the gross anatomy of the lung, we arrive at the phospholipid bilayer of an individual alveolar epithelial cell. Anchored into this membrane is the full-length RAGE receptor. Crystallographic data reveals 3 functional segments: a highly charged cytoplasmic tail, a single hydrophobic transmembrane helix, and an extracellular region composed of 2 C-type domains and a singular V-type domain. Here at the top of the receptor, the V-type domain serves as the primary interaction site. It contains 2 essential N-glycosylation sites that allow it to recognize incoming danger signals. Most immune receptors require highly specific pathogen signatures. RAGE operates differently. Its binding surface is highly permissive, allowing it to receive and lock onto entirely different molecular structures. The first major class of these danger-associated molecular patterns are advanced glycation end products, which form and accumulate rapidly under hyperglycemic stress. The second class includes the S100 or calgranulin family of calcium-binding proteins, which act as potent drivers of acute inflammation. The third is HMGB1, a nuclear protein that turns highly inflammatory the moment it is released into the extracellular space by necrotic cells. Because of this structural permissiveness, RAGE acts as a universal funnel. It translates wildly diverse forms of tissue stress into one unified intracellular signal. That signal begins below the lipid bilayer, at the receptor's short, highly charged cytoplasmic tail. Signal transduction requires a specific docking event. An intracellular effector molecule named diaph-1 attaches to the tail, propagating the signal downward. This docking activates the mitogen-activated protein kinase pathways, specifically ERK12, P38, and JNK, which cascade rapidly towards the nucleus. These kinase pathways converge on NF-kappa B, the master transcription factor for inflammation, which crosses the nuclear membrane and binds directly to the cell's DNA. Once bound, it induces the transcription of a massive wave of pro-inflammatory cytokines, specifically TNF-alpha, IL-6, and IL-1 beta. But the AGER gene, the gene that encodes RAGE itself, contains its own NF-kappa B binding sites. Activating NF-kappa B upregulates the creation of more RAGE receptors. Hundreds of new receptors rapidly sprout across the cell surface, ready to bind more ligands. In a high ligand environment, this genetic loop ensures the inflammatory signal never decays. It converts an acute biological stressor into self-amplifying tissue destruction. Pulling back to the physiological level, we find that alveolar type 1 epithelial cells in the lung express uniquely high basal levels of RAGE. When a severe viral infection like SARS-CoV-2 causes initial cell lysis, a massive wave of danger signals floods the alveoli. RAGE does not process these signals in isolation. It physically captures complexes of HMGB1 and lipopolysaccharides and drags them into proximity with neighboring toll-like receptor 4. This chart shows the result of that crosstalk. The synergy between RAGE and TLR4 produces an explosive amplification of IL-6 and TNF-alpha that dwarfs what either receptor could generate alone. RAGE also engages in a secondary mechanism of transactivation through the AT1R receptor. SARS-CoV-2 infection downregulates the ACE2 receptor, causing severe accumulations of angiotensin II in the surrounding lung tissue. This accumulated angiotensin II binds to AT1R, which then forms a heteromeric complex with the cytoplasmic tail of RAGE. This forces RAGE to fire continuously, even in the absence of its primary ligands. This hyperactivation drives a convergence of inflammatory cell death known as panoptosis. As these cells rupture, they spill even more danger signals into the microenvironment. On a macroscopic scale, this manifests as widespread endothelial dysfunction, catastrophic vascular permeability, and the formation of thick hyaline membranes inside the lungs. Multi-organ failure in ARDS is the inevitable result of RAGE-orchestrated crosstalk, stripping away the tissue's structural integrity. In the intensive care unit, this molecular destruction presents a specific clinical footprint. The body attempts to regulate this using a natural endogenous decoy receptor. Enzymes, specifically ADAM10 and MMP9, target membrane-bound RAGE. They cleave the extracellular heads off the receptors, releasing them into the surrounding fluid. This cleaved segment is known as soluble RAGE, or sRAGE. It circulates in the plasma, acting as a sponge to neutralize extracellular danger signals before they can bind to intact receptors and trigger inflammation. This scatter plot shows a vital clinical metric. Massive spikes in plasma sRAGE demonstrate a strong positive correlation with ARDS mortality. The concentration acts as a direct surrogate marker for the scale of alveolar cell destruction. Paradoxically, in chronic conditions like diabetes, circulating sRAGE levels are notably low. This absence indicates a dangerous failure of the body's inflammatory control mechanisms. Monitoring sRAGE levels is a direct, real-time measurement of the physical disintegration of the epithelial barrier. Because RAGE drives the hyperinflammatory loop independently of viral replication, it is a clear target for host-directed therapies. This table maps the 2025 clinical pipeline for RAGE inhibition, detailing how therapeutics target specific receptor components. The leading small molecule antagonist, aziliragon, works by securely capping the V-type domain, physically blocking incoming ligands. Originally evaluated in Alzheimer's trials, its safety profile allows it to be repurposed. It is now tested to block ligand-mediated immune storms in COVID-19 and to suppress myeloid-derived suppressor cells in pancreatic cancer. A highly targeted genetic approach is currently in phase 2 trials, ARO-RAGE. It is an RNA interference therapy designed to silence the AGR gene. It intercepts and dissolves the mRNA strand inside the cell nucleus, halting receptor synthesis entirely. This directly breaks the pathological positive feedback loop. It starves the cell of RAGE receptors, neutralizing the threat regardless of how many inflammatory ligands are circulating in the blood. Disrupting the RAGE-ligand axis addresses the central engine of the inflammatory surge, providing a pharmacological path to halt the self-amplifying cycle of the cytokine storm.